Hi and a very good day. I am Dilla and my group mates are Maisarah, Farah, Emilia, Hanis and Yuvaraj. In this video, we'll explain about one of the downstream processing method, which is precipitation. Protein in fermentation broth is separated from other components which will increase its concentration and purity through protein precipitation. Two commonly used methods are adding organic solvent and salting out. The first method, which is precipitation through adding organic solvent, is carried out at a temperature below -5 degrees Celsius to avoid denaturation of protein. Ethanol, acetone, and propanol are usually utilized in this reaction. When organic solvent is added, reduction of dielectric constant occurs, resulting to stronger electrostatic forces between protein molecules and decreases protein solubility. The solubility of protein as a function of the dielectric constant is denoted as ds as shown in this equation, as ds decrease has not decreased too. Observing this diagram, the water hydration shell originally surrounding the protein is now replaced by alcohols which favor binding to each other, allowing protein to aggregate and precipitate.
Some limitation of precipitation by organic solvent is in terms of efficiency, it is less efficient for hydrophobic membrane proteins. The temperature must be very low, around 0 degrees C, to avoid denaturation. It also causes heat evaporation from addition of water due to hydration of solvent molecules. The protein concentration range must be around 5 to 30 mg per ml. The protein precipitate can be left too long in the solvent, and the salt concentration should not be high, which must be around 0.05 to 0.2 molar.
Now, I would like to continue with the industrial application that uses organic solvent to precipitate the protein. One of the examples is the process in industry to treat the wastewater. The sludge in the wastewater contains many types of protein. Protein recovered from the sludge waste stream will reduce the organic load and reduce the treatment cost. The recovered protein can be sold as a commercial product, such as animal feed, adhesive material, and in manufacturing bioplastic. Ethanol and acetone are normally used to precipitate the protein. Addition of solvent to the solution that containing protein will reduce the solvatic power of the water for a hydrophilic charged molecule. Aggregation that occurs in that solution due to the electrostatic and the van der Waals forces. This process is known as sustainable circular bioeconomic process development, which should be implemented in all the industries. So these are the detailed process how the process occurs. So next is example of purification of zinc paint using a milk clotting enzyme and Tibetanol.
Now I will proceed with precipitations by salting out. Salting out occurs in the aqueous solutions of high ionic strength that reduce the molecule solubility, causing a certain protein to precipitate. It means that the addition of high amounts of salt in aqueous solutions causes the solubility of a protein to decrease and lead to precipitation. Thus, salting out can be used to separate the protein based on their solubility in the presence of high concentrations of salt. Salting out relies on the changes in solubility based on ionic strength. Therefore, it is crucial to determine the ionic strength of aqueous solution before the salting out processes, as it has the relationship stated in the aqueous solubility of aqueous solute.
The advantage of salting out is that it is an effective means for initial molecule purification, that it helps remove protein that easily aggregate, and then it minimizes the unfolding stress to protein encapsulate and does not require the presence of a purification tag on the protein. However, it does come with the limitation that it lacks the ability to for precise isolation of a specific protein, which means that it cannot be a standalone matter. Contaminants often precipitate with the protein of interest, and the protein at the end of the product will be in a high concentration of salt.
I will move on to the industrial applications of salting out precipitation method. Firstly, extraction of DNA from human-coated cells. Initially, saturated sodium chloride is used to precipitate other unwanted proteins, followed by centrifugation, and the supernatant-contained DNA is added to polypropylene tube before absolute ethanol added. The DNA precipitate then separated and quantified. Secondly, is to measure relative surface hydrophobicity of bacterial cells, which was applied to E. coli strains with different surface protein antigens. This is done by adding different concentration of aminosulfate salts, and the order of precipitation will measure the hydrophobicity of the protein. Lastly, fabrication of hierarchical porous carbon HPC for additive storage, catalyst, and water purification. Different concentration of sodium chloride used will affect the salting out state of egg white protein, changing morphology and pore structure of porous carbon products. For example, implementation of HPC in lithium-ion batteries. In conclusion, proteins can be separated from other components by precipitation using certain salts. Some implementations require further treatment to increase the purity of protein. Two major methods used have their own advantages and disadvantages based on the condition and material involved in the separation.
That's all from us. Thank you.